Nice to meet you. Welcome to the afternoon session of Seoul International Biomedical Conference 2022. And I'm your MC for the afternoon. I'm Lee So-yeon. I hope you have all enjoyed lunch. And the theme of this conference is Post-COVID-19, New Future for Bioindustry. First, we have a special lecture for you. For Global Startup Case Study, Director Maria Plodinek from Artidis will be making the next special lecture. Please welcome her with a big hand.
It is my great pleasure to start this afternoon session immediately after lunch. I hope you are all awake. I will do my best to wake you up in the next 20 minutes. So it is my great pleasure to visit Seoul from Switzerland and to present our startup and also how we are addressing some of the problems or topics that were discussed this morning. In this first slide, actually, I think it summarizes already the themes and topics that were so beautifully presented in the morning session that were by far accelerated by the latest COVID epidemics, from the adoption of telemedicine to decentralization of the healthcare. There were a couple of talks addressing that, data access, interoperability, and how can we use those learnings from the COVID pandemic to actually improve care to our patients, to improve the entire workflow of the patient journey, have a more adaptable, resilient care in the future. There was a lot of learning on that in the pandemic, and important aspect, invest in data infrastructure. I work in oncology. Oncology is the area that was obviously not in focus as we had to deal with the more imminent dangers of the pandemics, but as a consequence of the COVID, there is a huge number of cancer patients that were not diagnosed in time, number of procedures that were postponed, and this opens up a lot of, obviously, questions and issues to be solved, but also to use innovative approaches to tackle some of those aspects. For example, latest data show that in the NHS system in UK, up to 93% of patients due to backlog as a consequence of COVID, did not get in time the appropriate appointment with their physician or the right time to get to their cancer diagnosis. Cancer diagnosis is something that is very much focused around centralized lab approach. It takes time. It's also not always in today's assessment accurate enough to provide best treatment options for cancer patients. So how can we disrupt the cancer care and use some of the learnings that we have also seen in the pandemics? Artidis, a company, a startup that I represent today, is really working in this space, trying to change how we diagnose and treat cancer patients. So some of the things we challenge is really, does tissue biopsy processing requires a central lab? Can we move that to patient bedside? How do we change and reduce patient anxiety, speed time to treatment, but even more importantly, get the right treatment the first time? And then as a consequence of that, optimize the entire patient journey and allow the different data sources, so combining technologies, hardware and software, allow to effectively merge different data that we collect in clinics. Our technology is already clinically validated. We are continuing to do large clinical programs, which I will present today. It is focused on patient bedside approach, and I will describe in the next slides also more closely what we do. But before going there, I want to emphasize, and now thinking about my introduction also about the consequences of pandemic, We often think that a lot of things in oncology space have been solved. It's not the case. We have great advancements, especially in the areas such as breast cancer, in terms of early screening programs, and also in some treatment options. However, it is very important to say that despite number of early diagnostic procedures and early screening programs, 85% of women at time of diagnosis actually have invasive disease. 30% of those will go to metastatic stage, and 71% of the women with metastasis will not survive past 5 years. In lung cancer, which is actually a #1 cancer, we see even more poorer results. And I choose those two areas because these are the areas where we work and try to use our technology to improve the patient outcomes the most. But our technology is not limited to a particular solid tumor, and we can work across different applications. One of the areas in the treatment that we target from the diagnostic biomarker approach is actually neoadjuvant therapy setting. Neoadjuvant therapy setting is an emergent therapy setting, and in some areas such as lung cancer, it actually is even moving to stage 1, so very, very early cancers, where we treat disease systemically before any surgery or radiation. Meaning the patient will certainly receive a biopsy, and there will, in any near future, there will be no replacement for core needle biopsies or fine needle biopsies, because we need to get the information about the cells as well as their environment. And liquid biopsies can be used as a screening tools, but they cannot provide information about tumor microenvironment. So once we do that biopsy procedure, we start to treat patients. And we treat patients based on the information we have today. That information is not sufficient, and 60% of the patients will actually progress after the first line of neoadjuvant therapy. So how can we use new technologies, new approaches to improve that aspect of patient care, and at the same time, coming back to what I mentioned at the intro, focus on providing the best care at patient bedside. Here is just shortly the numbers for breast cancer. As I mentioned, despite advances in treatment, there is no significant improvement in the overall survival for the past decade. So these are numbers presented this year at ESMO, and I think this is very important to have in mind when we talk about oncology. There is a lot that has been done, and there is even more that we still need to do. So a little bit about Artidis. So what is Artidis doing? Artidis technology is a bedside technology. At the heart lies a device, which details I will explain, is positioned in interventional and radiation oncology suites, and it measures physical characteristics of tissue at the molecular level. That information serves to diagnose disease and optimize treatment, and in conjunction with other clinical parameters, actually we can optimize the entire patient workflow. A very important part of Artidis is that it's a platform. So as I said, it integrates the device insights with other information within our digital tool called ArtidisNet. ArtidisNet is a fully fledged digital platform that allows to connect directly into EHR system and pull different types of information, being histopathology, imaging, genomics, in a way that the data can be analyzed in conjunction. This is very, very important. Today, we have opportunity to have access to huge amount of clinical data. Mostly, these are retrospective. Mostly, different information, even for the same patient, is not available. And so the longitudinal information about the patient progress and disease course is missing. So having the setup where we can look, monitor, and analyze our patients, as much as from the technical perspective sounds easy, it's actually some of the most difficult things to do. And we have worked for a number of years to ensure not just on the technical side, but also on the legal and regulatory side, that we can connect our platform to the electronic health record system. One of the examples that I want to use is our collaboration with MD Anderson Cancer Center, which is the #1 cancer center in the world, where we are currently integrating our platform into their EHR system for our clinical trials. Our technology can do two main things: diagnose disease from tissue biopsies immediately post-excision, and second, use that information to personalize the treatment. So how do we do that? One of the most important aspects of our technology is this nanosensor that you see on the right-hand side. The basic scientific principle is based on the technology called atomic force microscopy. It was invented in 1986 by IBM in Switzerland, so it's a Swiss invention. And for the component, so not the one that measures biological tissue, but can measure conductivity of materials, the team actually also received a Nobel Prize. So how do we use that technology? So that sensor is very, very sensitive. It's only 20 nanometer in diameter. That allows us to physically probe characteristics of all different types of cells and cell surrounding elements such as matrix or interstitial fluid pressure, anything that contributes in addition to cellular components to tumor growth and progression. That information we use to say, is the lesion benign or malignant? Exactly what type of lesion we are seeing. Is it fibrosis? Is it hyperplasia? How aggressive it is. And I will explain how physical characteristics tell us in a very easy way how aggressive the tumor is. And then last but not least, very important for treatment planning, the degree of physical changes in tumor microenvironment. If the tumor microenvironment is not favorable, any kind of therapies, but our focus, as I mentioned, is neoadjuvant chemotherapy and immunotherapy, will not reach the desired tumor site. So understanding the mechanics of it and how to manipulate it is key to actually have improved therapy outcomes. One might ask, so, there are so many biomarkers. We can do genomics, thousands and thousands of genes. We can do proteomics. So why would physical characteristics matter? Well, they matter for a very, very simple reason. Now think about it. How does a tumor metastasize? If a cell cannot extravasate and leave the primary tumor, it won't metastasize. And if it doesn't metastasize, the patient doesn't die. So the physical characteristics, ability for the tumor to deform, to be soft enough to leave the primary site, is critical. It's the part of the disease that actually kills the patient. That's how cells from lung go to bone or liver and so on. And the other aspect is that environment around the tumor. If tumor microenvironment is not heterogeneous, mechanically heterogeneous, then medication can, or activated immune cells will easily find their target at the primary site. So understanding mechanics of that actually in a very easy way provides those two critical information. How aggressive and how will it respond to particular therapies? Here is an example of tissue imaging we have done in collaboration with MD Anderson, where we really identify checkpoint inhibitor sensitive versus resistant patients, and then use that information to actually change radiation therapy in a way to open tumor matrix. Importantly, Artidis measures various types of physical characteristics: adhesion, elasticity, and also deformation, or what we call viscosity. So that complex information in one biomarker will describe all the characteristics of tissue in place. How do we implement it in clinical workflow? In a very easy way. You see up standard of care, and bottom is adding Artidis. So you directly take a biopsy using ultrasound guidance or MRI, and then immediately at patient bedside, the Artidis measurement can be performed. All this different type of information can be delivered to patient and to the clinician. So for the patients that actually don't have malignant disease, they have that information immediately after 1 hour. They don't need to wait days or weeks to get this information. And then, of course, cancer patients will continue in a clinical workflow and get their standard histopathology and genomic biomarkers. This is very, very important. So we do not replace what is already there. We provide information that we don't have today. And another very important part is that ARTIDIS does not destroy the sample. So you can actually use the same sample for any downstream analytics. Similarly, is in lung cancer, with the difference that there we use more frequently the radiation, different types of radiation modalities to change tissue properties. Our company, and that was the kind of the theme about the global perspective, we work with partners around the world, in Europe, mostly in Basel, Switzerland, in Germany, as well as in Spain, Barcelona. And US, we, among others, have a strategic collaboration with Texas Medical Center and specifically with MD Anderson, what I mentioned earlier. And here we go. And as a part of that strategic alliance, we actually work together in a very specific manner to tackle the entire value chain, from diagnosis to changing the treatment, and then from the clinical validation to actually go into commercialization. That is very, very important because once we have clinical data that are positive, how fast we can bring that to a real clinical setting where a larger number of patients can benefit. This is a study that just launched, so this is a premiere. We have worked with Texas Medical Center for years, but the strategic alliance just started in September as a continuation on large number of tumors and a larger scale that will look at longitudinal data from patients. This is important. A lot of data points from the same patient over time to see how our nanomechanical signature can be used to change the therapy outcomes. Our focus is in immunotherapy and chemotherapy in neoadjuvant setting. And finally, we are currently working across United States to establish our technology in the next years, initially in those 11 PPS exempt centers, some that are familiar with US healthcare system. These are very, very important healthcare centers that allow to bring new technologies early with favorable reimbursement conditions, and also they provide the right volume. That's why it's also my pleasure to be here in Seoul and talk to some of the colleagues here that are interested in maybe adopting such technology in Korea, because in terms of the payer system, in terms of the healthcare system and the volume, in thinking from the global perspective, this is very, very interesting to Switzerland. For example, just 3 hospitals in Houston see 20,000 breast cancer patients a year, which is the same amount as entire Switzerland. So for us, it is very important to act and work globally. And with that, I will finish with a summary about our company. I'm happy to discuss with you in one-on-ones in the next minutes or coffee breaks. And warm greetings from the Basel team to Seoul. I hope I will next time bring some of my colleagues together here. And I wish you a wonderful afternoon. Thank you very much.
네, 박수 부탁드립니다. Thank you for your presentation.